Introduction. Lepidoptera is an order of winged insects including butterflies and moths. There are more than 120,000 species of Lepidoptera recorded. The word of Lepidoptera comes from the Latin word equivalent to the lepido and from the ancient Greek words lapis and pteron. Therefore, it stands for insect with scaly wings. The phylum of this order is Arthropoda, under the class of Insecta. Lepidopterans live in almost every terrestrial habitat. Butterflies and moths are flying insects that fill the air. Lepidopterans feed on fruit substance like nectar, but some of the species feeds on other substance like animal blood and tears. Adult Lepidoptera can be recognized by the wings dense scale cover, which means give more or less complicated color pattern and design. Reproduction of Lepidoptera usually sexual with separate male and female individuals. Adults, male and female, will come together and recognize each other as a member of the same species by advertising with bright color, sounds, and odor. Butterflies and moths have complete metamorphosis, which means that they have four distinct stages in their life cycle, starting from egg, larva, pupa, and adult. The metamorphosis from larva stage to the adults occurs during a pupal stage. The objective of this field study analysis is, first, to study about diversity of Lepidoptera in UiTM Tapa. Second, to observe the population of Lepidoptera species in UiTM Tapa. The third one is to determine the appropriate technique in capturing Lepidoptera species.
Next, I will explain about methodology. First of all, the type of sampling that we use in this field study is random sampling. Random sampling is used to choose an unbiased sample. In each region, every segment of the region must have an equal opportunity of being selected. Next, the sampling technique that we use in this field study is capture-recapture technique. Capture-recapture technique is the method involved taking two separate samples from the population. The taken sample help estimate the population size and can be used to gather more information like survival rate and recruitment over time. In this field study, we had captured the sample and released it back to its habitat to avoid any harm to their species. For the equipment that we use in this field study is sweep net and cup. Last, number of replicates. There are two locations that we use for collecting the sample. The first location is Gamma Block and the second one is Faculty Building. Both of the location we do 2 times sampling. Next, we will show you the process of collecting sample that we do for this field study.
And now we will go to the analysis of the findings and results. In this study, we use a capture-recapture method in two locations around UiTM Tapa. The first location that was studied is the Gamma block, and the population of Lepidoptera captured resulted to a genus of Hydrilla, Lysa, Borbo, Junonia, and Amata. The number of each genus captured is 1, and the total population is equal to 5. The second capture in the Gamma block area resulted in total of 6 Lepidoptera, which is genus of Bertula, Tridripana, Lysa, Apipagis, and 2 of Spalgis. Moving on to the second location, which is the faculty building. For the first capture, we found population of Lepidoptera in the name of genus Anthotrypinae, Apipagis, 2 of Lysa, and 2 of Spalgis. This has resulted in a total of 6 Lepidoptera captured. Meanwhile, the second capture in the faculty building resulted in a total of 6 Lepidoptera, which is a genus named Phacelia, Ypitima, 3 of Lysa, and Daphnis. Next, to find the population density, we need to divide the total population by the total area in square kilometer. In this study, the total area used is in UiTM Tapa, which is 0.077833 square kilometer. For the area in the Gamma block, the population density for the first capture is 64 square kilometer, and for the second capture is 77 square kilometer. Meanwhile, in this area of faculty building, the population density for both the first and second capture resulted in 77 square kilometers.
During the process of collecting the Lepidoptera species, we find that the population is bigger at the faculty block compared to the Gamma block. This could be identified since faculty block is surrounded with flower plants and Gamma have less flower plants. As we know, Lepidoptera species or moth and butterflies are more attracted to flower and urban light. In UiTM Tapa area, the most genus that we found is Lysazampa. Lysazampa is a moth with a tawny brown wings with dark brown or black bands that created a striking contrast. These species are more attracted to urban lights. In this field study, we use capture mark recapture sampling technique. This technique requires us to capture the sample with our equipment, which is sweep net and cup. Then we release it and later recapture it again on the other day. By doing this technique, we are able to calculate the species population in UiTM Tapa. We done this technique twice to gain more accurate results. Somehow, there is another thing that can be improved, which is to mark the species with a non-harm ink so that we can do a population calculation precisely. Lastly, in our finding, we observe that there is a few species that is belong to alien Lepidopterian species, which is the Geometridae, Hydrilla, Lycaenidae, in our finding is Spalgis, Nymphalidae, which is Iptima and Junonia, and lastly is Crambidae, which is Epigagis and Accenthropinae, which is mostly a native species in Europe.
So, to conclude, this field study conducted at UiTM Tapa aimed to investigate the diversity and population of Lepidoptera species. The study utilized random sampling and capture mark recapture technique to collect data across two locations, which is the Gamma block and the faculty building. The result indicate a higher appearance of Lepidoptera species in the faculty building compared to the Gamma block, likely due to the presence of flower attractiveness. The most prevalent genus observed was Lysazampa, which is known to be attracted to urban light. The study concluded that the capture mark recapture technique proved effective in obtaining accurate results.
Here are some feature recommendations for our field study analysis. The first one is sampling strategy. Use a systematic sampling strategy to ensure representative data across different species of Lepidoptera. The second one is long-term monitoring. Use a long-term monitoring program to track more Lepidoptera species. This can help in identifying more species in different spaces in UiTM Tapa. By applying this recommendation, we can get more accurate data for our field study analysis.